So hi everybody, I'm Nancy Rabalais. I'm the director of LUMCON and also one of the research professors. I've been here since 1983, and before that I was at University of Texas Marine Science Institute in Port Aransas, and then before that in Corpus Christi, where I began my career 2 years in a junior college, 2.5 years finishing up at a regional university, 2.5 getting a master's degree at a regional university, 6 months working as a naturalist at Rabalais National Seashore, and then at the Port Aransas Marine Lab, and now here. And one of the things that is behind me all this way is the ability to recognize differences in species, which is what we're going to be doing on this workshop. So I began just by going on field trips and counting the fish in the trawl and taking mud samples and counting the animals in the trawl, and I got a job at Port Aransas in 1976 where I spent many days at sea dropping a big box core to get the sediment up and count all the little worms and things in there to look at the community of the South Texas Outer Continental Shelf. And I continue to do a lot of that work here at LUMCON, and we're actually still doing some of it. So I put this talk together for the non-scientists to the well-informed scientists. So hopefully I've hit the right thing here. So the whole talk is, what does biodiversity tell you? But you have to know what biodiversity is first. So we're going to look it up in the scholarly Wikipedia. So Wikipedia tells us about this. So what Wikipedia tells us is that biodiversity is the degree of variation of life, and this can refer to genetic variation, which would mean our DNA, and species variation, which would be a killifish versus a croaker, or ecosystem variation in area, biome, or planet. Well, not so fast. It's really just species variation, which is how many species do you have in an area. But more and more, that definition of a species is done by genetics. So my classic training in taxonomy, where you learn whether it's got 3 hairs or 5 hairs, is not, it's been superseded in many sorts of taxonomy by the genetic makeup. So in biology, a species is a group of individuals that cannot breed, that cannot produce living offspring by breeding with another species. But then there's this confusing thing about subspecies and then the genomics. So here's 2 subspecies in the Gulf of Mexico. These are stone crabs. Anybody ever had stone crab claws? They're really good. I worked on both of these species when I first came here, and their larvae, to see if their larvae had differences in their ability to tolerate changing salinity. Because the Menifee mercenaria from Florida lives normally in higher salinity waters than the one here in Louisiana that lives in lower salinity waters, although you'll find them all the way offshore on the platforms. So, but they do overlap in the area of Pensacola and do generate and are able to breed. So some people say these aren't really true species, but then the people who worked on the taxonomy, Austin Williams and Daryl Felder from ULL in Lafayette, definite color variations and definite behavior variations between the two. And the one in Louisiana is called dormir, which is French for sleep, because the stone crab here is much less aggressive, and you can hold it sometimes without getting pinched. And while we were raising these things, I needed their larvae. So when we were messing with them in the lab, if they got onto your thumb, you couldn't make it autotomize its claw and fall off. So you just had to grin and bear it until you could get somebody to help you get the claw and get your thumb out of it. So what I found is that the larvae of these are much more able to stand a wide range of salinities than these. So they are separate species and they're not separate species, which is why Bird called this semi-species, but it's really a subspecies. The name of a species is composed of the generic name, the species name, the people who identified it, and the year it was identified. So I want to show you the next one I'm very proud of. Whoops, not yet. Okay, this one. So, Dr. Nancy Rabalais in 1979 identified a new species in the Gulf of Mexico that looks like this. And the holotype is in the National Museum of Natural History. And I drew all these figures. And this scale right here is a millimeter. So you can think, okay, so 2 centimeters is an inch. This much is a centimeter. And you get 10 of those things in a centimeter, and you've got something about this big. But it was new, and I got to name it. Well, at first I was on one of these trips where you get the mud grab and then you pull the animals out, and you can see that I've never seen something like this. And so then I took a couple of them. I had several in a vial up to the National Museum of Natural History at the Smithsonian. And I went to see Austin Williams, who was one of the co-authors of this other species. And I said, I think I have something new. And so he put it under the microscope and he said, yes, it is. Because he knows everything about every crustacean in the world. He's a very famous, excellent scientist, who unfortunately is no longer with us. But so I, in my naive 21-year-old, said, well, what do I do now? He said, describe it. And I said, me? And so I did, and I'm very proud of it. I've done 2 other species names with Felder, the other one. And I am also the bearer of a species name of a nematode from China. So, you ever get asked that trivia question? So, that was and the other thing I love to do, actually, is think about the Latin and Greek roots of the name, because they describe what it looks like. So, Tenno is what? Does anybody know? Does anybody know what this is? Kelis. And tenni are teeth. So it's got a lot of teeth. And seps is, I'm trying to think of something in American. Encephalitis. Cephalopod is a octopus, but anyway, its head. So here's its head, and leva means smooth. So it's got a smooth head, because lots of them are armored, and it's got a lot of teeth on its keel. And Austin Williams helped me put the right endings on them so that they would be accurate, Latin and Greek, but I came up with the idea. So anyway, in the Gulf of Mexico, we have an area of low oxygen, which I'll talk about later. And these are the many kinds of some microbes determined by RNA genomics that make up the community of all the microorganisms in a water sample across a range of salinity from 0 to probably 4 or 5. And I can't even say these names. I can say Pseudonychia pseudodelicatissima, and I can say Tentacules leviceps, but I haven't learned all these words yet. And you can see, maybe, that some of these actually take up more of the total abundance when the oxygen is low than when it's not. Let me see, which is another one. And you can see that some of these take up more abundance when the oxygen is low. But this is a frequency of abundance of these different microbes in water samples across a chain of range of oxygen concentrations. And the importance of this is that these different microbes determine what kind of nitrogen cycling there might be, or what kind of carbon there might be, and it tells you something about how the ecosystem is functioning, because there's too much nitrogen out there, and if microbial communities can take up the nitrogen and change it, then we can have a more healthy ecosystem out there. So that's just two examples of the subspecies and the genomics. All right, so true or false? Terrestrial biodiversity tends to be highest near the equator, which seems to be the result of a warm climate and high primary productivity.
True.
I've heard a lot of truths. You're right. Okay, marine biodiversity tends to be highest along coasts in the western Pacific, where sea surface temperature is highest, and in mid-latitudinal bands in all oceans.
True.
Not exactly, because some of the communities in the deep sea are very diverse, but the animals are all very small. But it's an incredibly diverse community. Okay. Biodiversity in general tends to cluster in hotspots. True? I have no idea what this question is asking. Okay, so let's go on. So some of the features of habitats with high biodiversity, of course, are coral reefs and rainforests. And we worry about the cutting down of rainforests, and we worry about global warming and acidification in coral reefs and sedimentation and losing the structure and losing the biodiversity. So these are both areas that are in warmer climates, and the productivity, the growth of the coral and the growth of the forest is at a faster rate because it is warmer. And it's also warmer all year long. So the temperature is about the same, it's warm, it's high, and as long as there's nutrients to fuel the productivity, then they are going to have high biodiversity. And things like coral reefs and rainforests are also called bioherms, because the structure of the coral actually creates a habitat in which many other things can live. So if you increase the complexity of the habitat, then you're likely to have greater diversity. And all right, so over the years, people are interested in biodiversity scientifically, and we want to know, how many of what kind of sharks are still left in the ocean, how many whales of what kind are still in the Gulf of Mexico, how is the diversity of those whales changing over time, to just nice picture books. You wouldn't believe the Houston Atlas of Biodiversity, how beautiful this book is, and how many different species they have in it. You can have just a book that tells you all the Fishes in the Gulf of Mexico, probably missed some of them because there's always one more hiding out somewhere. And these books and others that you're going to see in the lab have something called a dichotomous key. So you start with, let's get very basic, has a backbone or doesn't have a backbone. So you've got a vertebrate and an invertebrate. Of these invertebrates, has 5 arms usually, or is vermiform, vermiforms. And then you go into has CT on the head, does not have CT on the head. And then you go, so you have a choice, a choice at every step in this key of how to figure out what this species is. And then a famous one for Louisiana, of course this is famous too, is George Lowry's book on mammals of Louisiana. And then other groups, so anybody ever been on an Audubon Christmas bird count?
What is it again?
An Audubon Society Christmas bird count. So 2 of us, and I know, yeah. I used to go, yeah, I know who your husband is. But I used to do these all the time in South Texas, and I get to go with my professor and get to access to lots of places where you couldn't normally go, like on the King Ranch, where we knew we'd find a great horned owl or something. So you go out for a 1-day period with your binoculars with a group of people with pads, and you count every bird of every type that you see. And people have been doing this for decades. And so it's given us a long history of how bird diversity has changed in various areas. So that's another way to get a species list. All right, here's the official scientific way to determine biodiversity. The easiest one is species richness. How many species are in an area? Okay, how many species are in this room? Probably 1, except for there might be things crawling around. Okay? But if there were things crawling around with us, of course you got all kinds of bacterial flora too. Don't forget those. You can come up with an index. This one's called the Shannon-Weiner Diversity Index that calculates how diverse the community is. It can be very diverse or not diverse. The other one is a Simpson's Diversity Index, and it's a lot simpler because it doesn't have a log function in it. You get this in the computer, it's really easy to do, but if you're doing it with a little hand calculator, it gets tedious. This one's a little bit easier. But total number of species is usually what people rely on. So what does biodiversity do? First of all, it's a symptom of a healthy ecosystem. And therefore it helps maintain ecosystem functions like cycling of carbon and nitrogen or provision of habitats for other species. It's also going to be the source of recruits. For instance, if a coral reef in one area has been devastated for some reason, and there's a very healthy coral reef somewhere else with lots of species, the healthy one could provide the recruits in the larval forms that eventually get over to the one that's not in good condition and help it repair itself. Stability and resilience, the more complex an ecosystem is and the higher the diversity, the more capable or the better able the ecosystem to respond after some sort of devastating illness or hazard or storm or human beings or things like that. So diversity is very important. All right, in the Gulf of Mexico, this is not a hypoxia lecture. So there's an area of low oxygen caused by river freshwater and nutrients that leads to an area on the bottom where biodiversity is diminished, and adjacent areas where biodiversity is quite complex. And it's the result of excess nutrients from the Mississippi River affecting the Gulf and leading to these areas of low oxygen. And these areas are mostly in the spring through the early fall. They cover large areas. This is 22, this is probably 20, yeah, this one is 20,000 square kilometers, up to 22, which is the size of the state of New Jersey or Massachusetts. And so they're large. They're from very near shore to further offshore, from about 4 meters near shore to 45 meters offshore. And when the definition is the oxygen less than 2 milligrams per liter, and if a trawler puts a net over and pulls the net, there are no fish or shrimp that normally live on the bottom come up in that net. So when you consider an area this large, that has huge implications for fisheries. So, like I said, I grew up looking at the mud. So, some of the consequences of this low oxygen is mass mortality, doesn't happen that often, a decline in a catch of brown shrimp over decadal averages, and then the animals that I love so much that live in the sediments, like this brittle star, start to not have enough oxygen, so they come up. They're normally under the sediments with the tips just filtering, and then they come up and be on top of the sediment and move around. The spider crab is what we affectionately call our smoked spider crab, because it did not survive. This polychaete, which if you count the different kinds of CT and the changes of them over the length of their body, you can figure out what species it is. Gotta love them. Okay. And then, so this one is so stressed it's up on the top of the sediments. But this animal and this animal, still being there means that no fish are getting in there to feed. This is a very dead spionid polychaete with sulfur bacteria starting to form around it. So this is very anoxic, low oxygen water. So what happens in the Gulf as you go from highly oxygenated to very low oxygenated is, first of all, you start to get a decrease in just the total number of animals. And then you start to lose things, like the fishes will start to move out. Some of the motile epiphytal, like brown shrimp, will move out. But when you get below 1.5, all of those are gone. And then you start to see the larger infauna, like the brittle stars, showing some stress. And then when you get down to these values, you start to lose the polychaetes and the things that live in the mud. And these are very important because they are the food source for these animals. That when the oxygen goes away, they come in to feed, and there's not as much food anymore. And then when it's really, really low, you get the production of hydrogen sulfide in these bacterial vents. So just in one spot, the environment can change over a seasonal cycle back and forth along this continuum. All right, so how do we determine biodiversity in a mud grab from the Gulf of Mexico? First of all, we got to get our mud. Okay, this is a box core. This is our longer one. At the bottom, which you can't quite see, are some shovels that close it off. It goes down open, and when it hits the bottom and you pull up on the cable, the shovels will close, and you'll bring it up, and you'll have a box core full of mud, which you can then subsample for. This is looking straight down on the mud, which you can then subsample for different things. You might want to do an experiment looking at nitrification. You might want to count the worms. And you might want to look at the bacterial community. And very often we take samples just by hand as well. And so this is a very low oxygen environment, black sediments, which would indicate probably not much living there. Okay, so we bring all this mud into the lab. We sieve it on a sieve, a screen, usually down to 0.5 millimeters. And then somebody, it's not always me, we had to, my technician who does this mostly wasn't here when I took this picture. But out of that sample, we've got to find all the animals and classify them as to what species they are and count them. And to do that, all these books behind me, right here, are books like The Mammals of Louisiana, except they're the polychaetes of Morocco, or the amphipods of Gayhead, Massachusetts, or something like that. And so then you can tell the difference in the species. Which one is this? This is a, can't read it. I worked on this project. So we saw a lot of these black dots here. Not many in the Central Gulf, and none over here. Possibility that we didn't know what we were calling it. But actually all these samples got looked at by a group of taxonomists who made all the names uniform and generated these keys. So it's an incredible body of work. Then you have to figure out what kind it is. Then you have to count them all. So this is a sample from a shell platform in about 20 meters of water, and it's called produced waters because we were looking at the effects of oil field waste disposal on the animals that lived in the bottom. And it's 1,000 meters away from the discharge. And these are all the major categories. And then these are all the specific names. And this is how many Lorraine at the time counted. So if you wanted to do a diversity index, we'll use the easy one. The easy one is how many species. How many species? There's 1, 2, 3, 4, 5, 6, 7, 12 species. Okay, that's different than 20 species or 1 species. Or we can take the calculation of the total number, 39, minus 1, and then the first one in the series is Paraprionospio pinnata. It's a very common worm, and it's this one. It's the number of that species multiplied by the number of that species minus 1. And then the sigma is the addition, so you add those all up and you come up with an index. You too can do this. And calculate. So, we use these values to tell us how an ecosystem, is it healthy or not, is it diverse? So out in the Gulf of Mexico there are we compared 3 oil platforms to see how the discharges around those platforms affected the organisms, but we also looked to see how the hypoxia, the low oxygen, affected them. So this one is closer to the Mississippi River, and these data are data from an oxygen meter near the bottom we deployed that every 15 seconds recorded how much oxygen was in the bottom water. So our DO of 2 or less tells us if it's hypoxic or not. So you can see that it goes hypoxic, but then it goes above hypoxia. And this is the month of June through October. And with passing of storms, cold fronts, you're going to start to get more oxygen. Okay, so out of that mud grab, and out of all those things, and out of that microscope, and into those little vials, we came up with this station. This is about 70, this is the number of individuals. So about 70 of these things in that sample in April. And it was made up of all these different kinds of groupings of species. And as you can see, there was some decline in the summer as it goes on, but it didn't always go below 2. So then we look at some of the specific animals. And our friend Paraprionospio pinnata, was really the only one that survived during the summer, and then it came down. This is another polychaete, but you can see that there were quite a few organisms here. So this is another station, and it's just offshore Terrebonne Bay here. And if you look at the tube, you can see that this station had very little oxygen for extended periods of time. And while the number of individuals in April, 120, of high diversity, as the oxygen went down, in this period right here, you can see that the number of organisms and the diversity of these different groups dropped dramatically. And then, and again, our good old friend Paracrinos piopenata, it survived. This one is Mediomastus. It's a very small worm, and you can have tons of them. And then everything else, including, let's see, this one doesn't have any starfish, but they all drop off. And then the following year, because it was such a severe year, the number of recruits that came back in were very low, and we had very low numbers for the whole year, and so the abundances were low. The oxygen, even though it went higher at times, was very low in the summer, so it dropped out again, and then some things it was a annihilation of the fauna, and no recruitment the following year. So that this area of the Gulf now is missing. These are groupings of, like, these are brittle stars. We just don't see brittle stars anymore. It's not just a loss of a species, it's a loss of all the brittle stars. Whether there's 6 species. So this ecosystem is not functioning. It's not turning over nutrients. It's not binding the sediments, or sediments getting resuspended. And when that happens, it provides organic matter for oxygen to be driven down again. So it just doesn't work. All right. If you want to have some fun, you can put all of these data into a computer with a program and find out how similar they are to each other. So these two stations, the Shell station, in winter and spring were very similar to each other. But they were not as similar to this as they were to each other. So this goes here. And then the fall sample is different from all of these. And you can do this, what's called a cluster analysis, to see how the stations are similar to each other based on how many numbers of species and the diversity.
Nancy?
Yes?
What would that cluster diagram look like if they were all the same? Would it be a straight line?
If they were all the same, they'd all come up to something like this.
Okay.
Straight across.
So it looked like a cone, kind of? Like
Yeah.
one line down and down?
Okay. And then you can really get fancy and do a discriminant analysis and see how they're related to each other through time. So basically it's taken that cluster and it's found out that when the oxygen is very low, these two stations in the summer are most like each other than they are these stations, which are fall samples. And that when the oxygen is intermediate, these stations are different than these, but different from these. And then when there's high oxygen, you can see that these stations, which are all in the same area, are more like each other than they are the stations further to the east. And so when this analysis was done, my colleague Dubravko Justic said, wow, look at this, and I sat there and counted all those things. I know exactly what these data were going to say. And I'm going to skip this. Our seasonally severe hypoxia is much worse than other areas in the U.S., like Chesapeake Bay. All right. Let's skip that. All right, so then we have this problem of invasive species. While this isn't and invasive species talk, they can have incredible effect on biodiversity. This is the zebra mussel. It clogs pipes, it creates an awful odor, it causes lots of damage to equipment, and it pushes out native mussel populations that, these pearl-generating mussels. So it causes a lot of problems, but it kills the native mollusks, which is going to decrease the biodiversity. The first one was recorded in the Great Lakes in 1988, and this is where they all are right now. And they're spreading like we expected along the intercoastal waterway, and these show up because they were trailered on a boat hull. So once loose, they're very hard to get back in. So this is our favorite local non-indigenous or invasive species, the nutria, which was brought in for fur bearing. They got loose and now they're all over North America. But what they do is they eat out the vegetation, which reduces the habitat not only for them, but for native species like muskrats and minks. And if you exclude these with a fence, you can see how much healthier the ecosystem can be with the structure and all the other things that are in there. Our favorite animal in spring, the crawfish, has been introduced into California for aquaculture and has gotten loose, and it's very aggressive, and it scares out or outcompetes the native crawfish, and also eats some of the newts, which are the little reptiles. And so it's devastating the biodiversity in some of the wooded streams in California. So we need to go over there and get it back over here. The other one is the lionfish. Beautiful Indo-Pacific fish, great in aquaria, and then one was let go, or escaped, it was probably let go, from an aquarium, and then one was spotted near Cuba, and now they're all over the place. And they outcompete the local fish, and so they're driving down the biodiversity. And we do have them on our offshore platforms here in the Gulf of Mexico. There are people who make it their lifelong goal is to wipe out as many lionfish as they possibly can in the Florida Keys. And there is lionfish sushi, lionfish tacos, lionfish, fried fish and chips. So they're apparently good to eat, and so there's a real effort to try to bring down the population. So this is sort of another example of loss of biodiversity. The Florida kelp forests are incredibly diverse. There's a lot of structure. There's a lot of animals, fish that live in there, things that live on the rocks on the bottom. And two of its natural organisms are sea otters and sea urchins. But in the 1800s, human beings hunted too many sea otters and reduced the sea otter population, which meant that its favorite food, the sea urchin, didn't have any predators, and we just got more and more and more and more of those sea urchins. And those sea urchins grazed on the kelp blades. So slowly but surely, they were able to kill the kelp forest. And the only thing left were all these sea urchins all over the bottom. But now with the comeback in the sea otter population, we're starting to get back to some normal functioning of this ecosystem. All right, one last thing, and you'll get the point here in a minute. Does everybody know what a rivet is? It's a little screw, but it doesn't screw, it gets pounded in. It holds boats together, it might hold some metal canoes together. It holds airplanes together, and there I am, going on a business trip with my laptop, trying to get something done at the last minute before I have to go into a meeting, And the rivets start popping off this airplane, one by one. And there's another one, closer to Dallas, getting close to Salt Lake City, getting close to San Francisco. So when is the last one too many? And that's one of the issues with biodiversity, because it holds our planet together. We affect it, and lots of other things affect it. And so I hope in your workshop you get to learn some more about the organisms around you and how they all fit together to make an ecosystem. So thank you all very much for being here. A little bit longer on that Shannon Wiener Diversity thing, but we've got time for questions, I think. Do we, Murder?
Yes, we do.
Okay.
Plenty of time for questions if anybody has one.
Processing.
Could you possibly discuss your current findings over the last year with hypoxia as we looked at the slides before, and understanding that in 1990 things were at that level?
It hasn't gotten any better,
Yes.
and it's related to the amount of nitrate coming down the river. And that hasn't been reduced because of big agribusiness.
Mm-hmm. What are the responses in the science community to your findings with all of the research concerning them?
It's consistent with similar problems around the world. There's no science issue here. It's a social and a political issue to try to make changes. And there's a lot of effort for sustainable agriculture and doing things differently, cover crops in the winter, deeper rooted plants, using less fertilizer. So there's a lot of solutions, but they're just not done on the scale that they need to be done. There's a lot of very well-intended farmers who are taking care of their land. But the farm bill and subsidies drive the farm economy.
Thank you.
We're bringing a group of kids down to Grand Isle on Tuesday, and one of the things we're going to have them do is go along the beach and try to look for different things on there. What type of, with the biodiversity and everything you were talking about, like what type of things do you think we should have to, trying to get these kids from in New Orleans in the city getting introduced to this? What would you suggest saying?
Well, in the daytime, the ghost crabs are going to be in their burrows if they're a smart ghost crab. And the things on the beach will be mostly birds, probably. But if you take a shovel and a piece of screen and dig up some of the sand under the water and throw it in that screen and shake it, you're going to hopefully be able to see lots of things that these birds eat. From little crustaceans to worms to amphipods. So you should be able to do a Christmas bird count on your wall.
Follow behind all the
Not in the snow. Yes.
So if we would reduce the amount of nitrogen coming down the river, how long would it take for the Gulf to recover?
There is residual nitrogen in the watershed and residual carbon in the continental shelf sediments. So it's not going to happen immediately. The estimate is 5 to 10 years if we completely stopped it, which is not going to happen. The goal is to get it down to a more reasonable size. The best example globally of this buildup and decline is in the northwest part of the Black Sea where the Danube River comes in. It's a continental shelf, very much like our continental shelf. And as nitrogen and phosphorus went up, the number of low oxygen zones went up, or the size of their low oxygen zone went up. And then the change in the Soviet Union and subsidies for fertilizers, the nitrogen and phosphorus started going down, and the number of, the size of the low oxygen step going down until it just didn't exist anymore. That was an economic collapse, which is not something we want. Yeah.
So how does or how would this research get to policymakers? Is there someone at Lumpcon who
We had our 30th cruise this last summer on this, but in 1994, a reporter came down named Joby Warwick from Washington Post, and he wrote all about it, and it made, in Washington speak, it made 2 inches above the fold on the Washington Post on a Sunday, and that caught the attention of Senator John Breaux. And Senator Olympia Snowe from Maine, when she says GOM, so there's more than one GOM. It's just the Gulf of Mexico, isn't it? She said, no, it's the Gulf of Maine too. And she was worried about harmful algal blooms. And she's Republican. She was looking for a Democratic co-signer of the bill. So Senator Breaux said, you put the word hypoxia in there, and we'll have a bill. And I went out and testified. And I've been in the middle of trying to keep it in the highlight for a long time since it generated some legislation. So, and we work in the watershed a lot. We work with sustainable agriculture groups. So my last trip was to Memphis, and then I had one to northern Mississippi, and then I'm going to Illinois in about a month. So we try to spread the word. We bring farmers down here. I've taken them out on the road, bring them back in, and one of them said something like, well, you're really not making this up, like. So there's a lot of people in Chesapeake Bay, the Great Lakes. The Great Lakes have the same problems with too many nutrients. And this last month ago, Toledo, Ohio, couldn't use the lake water for drinking water because there was too many phytoplankton toxins in it. It wasn't healthy. It was the same thing. It's agricultural runoff. So it can affect health, human health. It can affect living resources and ecosystem function.
And then even just you letting people know about the workshop and the work that we're doing, that's also another facet of getting the word out about what's going on. That's one of the reasons why Dr. Rabalais is here. To tell you
I could be sleeping right now.
We have donuts.
I'm going to go get some.
We appreciate it very much.
You're welcome.
Yeah.
We only briefly really talked about the oil spill,
Right.
and how that
Okay.
impacted
We're doing
species quality.
We're doing counts of species in the marsh sediments and in the subtidal sediments adjacent to the marsh. Those are very tedious samples because you get this much marsh muck and roots, stems, and the animals are this big, and so you have to sort through a lot of stuff. So what we found so far is that the species and the abundances in what we call our paired, controlled, and oiled and unoiled sites, 3 in Terrebonne, 3 in Barataria, and 3 in Eastern Barataria, is that if you have a group of 3 samples and you're looking for a mean, and the abundance is this high, and the variation might be this high, which means if you have a lot of samples like that, and you run statistics, you can't see any differences because the variability among those is just so great. So that measure is not telling us that, well, we're not finished with doing the samples because there are a lot of them. But that sort of information is just going to tell us that the marsh has a lot of variability in the number of species. The subtitle, we have something called a worm cam, which is a box that has a prism with a camera in it that you lower and put in the sediments so you can see what the animals are doing in the sediments. And we're taking sediment samples, smaller ones by person, and we're counting those, and those are easier to do. And those seem to be telling us something different with the ones that are adjacent to oil versus unoiled. And others, our group, and I haven't seen the numbers, are looking at the number of snails and mussels. And the mussel data indicate to me that they just didn't know how to find mussels when we first started, and now they're much better at it. So it's low and now it's higher, but I don't think that's real. And people are doing insect studies, loss of insects, low recruitment, loss of even more with Hurricane Isaac, and the sediments, the sediment chemistry in the oiled and unoiled. There's definitely still oil in oiled. It's covered up with sediments. In the summer, it still releases a lot of aromatic hydrocarbons, vapors, and it kills things. So you can, I used to torture fiddler crabs for my dissertation, but you can put crickets in traps out in the marsh over the oiled plots versus the unoiled, and the oiled ones are all going to die because the heat makes the fumes still come up. And those crickets, or katydids, or ants, or spiders, are the base of the food web that includes the seaside sparrow, which is having reproductive capacity declines in the areas that have been oiled versus unoiled. So it's, you know, you can go out there and look. It doesn't look like there's any oil, but there's still some effects.
Any other questions? If not, let's give Dr. Rabalais another round of applause.
Thank you all for participating. It's great to see so